So our second presenter is Professor Dennis Low from the Chinese University of Hong Kong. Professor.
Testing. So good morning. It's my honor and privilege to present in front of this distinguished audience today. I'd like to thank the RGC for funding our research. So today I'd like to talk about our effort to try to develop a universal cancer screening test. So over the last 15 years, my group at the Chinese University of Hong Kong has been exploring the diagnostic use of extracellular DNA which swims around in our plasma. So we're interested in the release of tumor DNA into circulation of cancer patients and also the release of fetal DNA in the plasma of pregnant women. As you'll see later on in my talk, working in both of those areas in parallel have given us some strategic advantage. So first, some of the preliminary background about our work on cancer DNA. Now for this, so it's been known for a while that during the course of its biology, a cancer cell will release DNA into circulation of cancer patient. But for this approach to work as a tumor marker, you need to find a species of DNA which is specifically associated with the cancer. And in this regard, some of our early work were focused on nasopharyngeal cancer, or NPC. And the reason is because almost all NPC cases in Hong Kong are associated with Epstein-Barr virus infection. And the EBV DNA can be found in the nucleus of NPC cells. So we postulate in 1999 that maybe during the course of its biology, an NPC cell will release EBV DNA into the circulation of cancer patient. And indeed, our work showed that the plasma of NPC patient is stuffed full of EBV virus, like shown on the left-hand side. On the other hand, if you have patients, subjects without NPC, then most of them are completely negative. So by use of this very simple approach, you have a sensitivity detection of around 96% and a specificity 93%. And also the level of this EBV DNA that you detect in plasma is related to the tumor stage. In that early stage disease, those in stages 1 and 2, have a median of 8 times lower level of EBV DNA in plasma than those who are in the advanced stage, those in stages 3 and 4. And so basically showing that what we're measuring in plasma is a reflection of tumor load we have on board. And furthermore, the higher is the tumor load, and so the higher is the level of EBV DNA, then the higher is the chance of those subjects dying of cancer. So this is a very powerful prognostication tool. And in addition, you can also use this plasma DNA to monitor the progress of treatment. Now, for example, the mainstay of treatment of NPC in Hong Kong is radiotherapy, which typically takes between 6 to 8 weeks. So you can see that if you monitor patients undergoing radiotherapy, even before the radiotherapy has finished, you can already see exponential decay in the EBV DNA. And you can even measure the kinetics by looking at the half-lives. So if you have a log of EBV DNA plot against time, you actually have a straight line. And you can see that different individuals have different half-lives. And actually, this data have been shown to be a reflective of the radio sensitivity of a particular tumor. And now, when you treat and finish treatment, and if the level becomes 0, then actually, by and large, the patient do very well. But if the level is between 0 and 500, then they do less well. But if it's above 500, then all of them die. And furthermore, you can also use this tool to screen for patients. For example, we're just in the middle of a study in which we're screening a group of healthy subjects in Hong Kong, prospectively done. So currently, we have screened 3,000 healthy Hong Kong subjects, and actually 5 of them was in stage 1 or stage 2 disease. So from all of the above, you can see that this plasma DNA test for NPC is almost an ideal cancer marker. But the problem is not all cancer are caused by EBV, and not all cancer are caused by viruses. So the objective of our theme-based research project is to see if we can develop a general universal cancer test, which can go beyond the confine of what I've presented to you just now. And in this regard, I'd like to see if we can get some hints from our other project, which was actually funded by the AOE scheme. So in that project, we're looking at the release of fetal DNA into plasma of pregnant women. And in this project, the most important goal is to try to see if we can develop a test for Down syndrome, which is caused by having an abnormal number of chromosome 21. So in 2008, we have developed a method whereby if we extract the DNA from plasma pregnant women, and then we do massively parallel sequencing of millions of DNA molecules, and then if we map those sequences back to the human genome to see from which chromosome each of those molecules is coming from. And then we can plot a proportional representation of each of the chromosomes in plasma. So we can have a histogram like that from every case. We can plot a distribution. And then, for example, in chromosome 21, I can compare that signal with a group of control and see if there's any statistically significant difference. So we use a Z-score. So if there's no difference, the Z-score will be around 0. But on the other hand, if there are more chromosome 21 sequences, the Z-score will shoot up. And indeed, when we first do this work, we find that if you have the cases in green and red, actually the Z-score is very high. And as you can see, those cases, the baby have trisomy 21. But on the other hand, if you look at those cases in blue and orange, then actually the Z-score is around 0. So those cases are normal. And this technology has now actually been validated in over 20,000 cases. And the sensitivity and specificity are both over 99%, and has been clinically launched since 2011. And now actually is available in over 15 countries, and 500,000 pregnant women have been tested, which is really quite remarkable for such a relatively complex test. But our question for this film-based research project is, can a similar test be developed for cancer testing? Now, so going into this, we know that this is going to be quite challenging, because for Down syndrome detection, you're talking about a whole chromosome which is abnormally represented. But whereas in cancer, any chromosome can be abnormal, and any bits of each chromosome can be abnormal. So we have to have a solution which is subchromosomal in amount. So what we've decided to do, instead of looking at chromosome level, we actually split the genome into 3,000 segments. And so each segment is 1 megabase in size, as compared with whole genome, which is 3,000 megabase. And then we sequence hundreds of millions of DNA molecules in the plasma of a cancer patient, and then we'll compare it with a group of control, and we'll use a Z-score approach like what I outlined to you just now for the trisomy 21. So initially, we start off by looking at liver cancer, because as Natalie has shown you, this is a common and important disease in Hong Kong. And this is a Barcelona stage A cancer. It means it's resectable, and the tumor size is about 6.2 centimeter. So if you look at the tumor tissue, this is what you get. So you can see that there's some areas which are amplified, those in green, and some areas which are deleted, those in red. So this is tumor tissue. So what we like to do is to see whether I can replicate that data by looking at the plasma. And interesting, by use of our technology, you can see that by sequencing the plasma in this patient, I can actually replicate most of those changes that I see in the tumor tissue. And after surgical removal of this tumor, then most of those changes have gone. So there's one case. And this is a second case, and also Barcelona stage A, means it's resectable, and this time the tumor size is even bigger. You can see this is a tumor DNA, and this is a plasma before operation and plasma after operation. And this is a third case, and very similar data. But on the other hand, if you look at chronic hepatitis B carriers without liver cancer, then the signal is very clean. You can see it like this. And this is second patient, and third patient, and fourth patient. And then we thought that would be nice if I was able to change this test into some sort of quantitative readout about what is the level of tumor DNA in circulation. Now, normally, if you look at our autosome, we have 1 copy from our father and 1 copy from our mother. And normally, those ratios should be the same, so 1-to-1 ratio. But in cancer, every now and then, in one of those copies, 1 bit of it might be deleted. Now, if that's the case, then you will have this imbalance of the 2 copies. So we reason that by measuring the level of this imbalance in plasma, we can calculate the cancer DNA concentration in plasma. Now, for example, in this particular case, we can look at the area with 1 copy loss, and then we zip them together, And lo and behold, you can see that in this case, in which the tumor size is 14 centimeter, just over half of all the DNA in plasma before surgery is from a tumor. And then after surgery, it has drastically reduced. And this is another case. The tumor size is a bit smaller. And then before operation, 7.6% of all the DNA in plasma is from a tumor. And after surgery, it's reduced to 2.7%. And this is a third case, 6.2 centimeter, and before operation is 5.6%. You also see that there seems to be a correlation between the tumor size and the level of tumor DNA. So bigger tumor has more DNA in plasma. And also, we decided to actually challenge the system a bit more. And over the course of this work, we've actually identified a woman who is a BRCA1 mutation carrier, who at the same time carries both a breast cancer and ovarian cancer on both of her ovaries. So we obtained the samples from the breast cancer and also 2 regions from the right-hand side ovarian cancer and 2 regions on the left-hand side. We call that A, B, C, and D. And this is a result from ovarian cancer. You can see that from those 4 sites, A, B, C, D, so basically the grossly genomic aberration can be seen across the whole genome in ovarian cancer. And similarly for breast cancer. But actually, the changes in the ovarian cancer are different from breast cancer, and each of those 4 sites in the ovarian cancer have the similar profile. So then we capture, this is ovarian and this is breast. So the question is, what happened in the plasma? So when you do the plasma before surgery, you can see basically it's a overlapping, a superposition of the signature from ovarian and breast cancer. And after surgery of both tumors, then actually most of those changes have gone. Now, if you look at the data more detailedly, you can see there's some areas in which one cancer have the change and the other doesn't. So let's see in this area, for example, in chromosome 6p, the breast cancer has a 1 copy loss, whereas the ovarian is normal. So basically, a signal that you see from plasma is originating from breast cancer. And by looking at this for each tumor and across the whole genome, then we're able to calculate, in this case, before surgery, 46% of all the plasma DNA is from the ovarian cancer, and 2% is from the breast cancer. So next, we become more ambitious. So we thought that, well, if the copy number aberration profile of all the 4 ovarian cancers are the same, but I really like to know if we can further dissect the contribution of each of those 4 sites in the plasma. So what we did is that we obtained the 4 sites, and then we sequenced the genome of each of those 4 sites completely. And then we group the mutations into a number of groups. So there are those mutations which are shared by all 4 sites. So there's just over 2,000 of those, so we call that group A, B, C, D. And there are also those mutations which are only present in the right-hand side, so we call that AB. And there are also those mutations which are only present on the left-hand side, we call that CD. And there are mutations which are private to only one site. And next, we're trying to hunt for those mutations in the plasma from the sequencing data and calculate their fractional concentration. And this is the result. You can see that the fractional concentration of the mutation, which are common to all 5 sites, represent about 46% of all the DNA in plasma. But on the other hand, if you look at those which are just on the right-hand side, this is 9.5%, and those on the left-hand side is 1.1%. And actually, this ratio is actually the relative size is consistent with the relative size of the tumor on the right-hand side and that on the left, being the right-hand tumor is bigger than those on the left. And then you can see that for the private mutation, the fractional concentration is pretty low. So it means that in one go, in one plasma sample, you can actually dissect the heterogeneity profile of this tumor. And this is actually a significant step forward from what people have been doing before, because prior to this work, if you look for tumor DNA in the plasma, you'll be looking at 1 mutations or a couple of mutations. So now, if I'm looking at this, you can see that how can you be sure that those single mutation or couple of mutation are representative? Because you might be measuring those concentrations, in which case, even if you go to 0, it doesn't mean that you don't have any tumor cell you have on board. So basically here would be one way to actually look at the tumor holistically. And also because of this recent interest in intratumoral heterogeneity, this is also very powerful ways for you to actually look at that in the plasma, because the plasma receives all the DNA from all the heterogeneous clones across the whole body. So in one go, you can do that. So we call this technology cancer genome scanning in plasma. So we published that in October last year. And 1.5 months later, Bert Vogelstein group from Johns Hopkins also published a very similar paper. And also early this year, a group from Cambridge and also a group from Austria have also published that. So basically now we have 4 independent confirmation that it's possible to scan for the cancer genome in the plasma. But the problem with this approach is that we're talking about 400 million reads per case, so it's pretty expensive. We calculate at the time when we did this work, it costs about 80,000 Hong Kong dollars to do 1 case. So this is clearly too expensive. So for our last year, we've been thinking about how can we cut down the cost. In this regard, we would like to look into the area of epigenomics. Now, so the genomics or the genetics side concern the sequence in the DNA. And the epigenomics is a biochemical modification of that sequence. Now, for example, as an analogy, the sequence we like if I do a building, which is the walls, etc. And the epigenomics will be how you decorate those walls, how you put in the furniture. So we're particularly interested in a class of epigenomic changes called DNA methylation, which is basically the addition of methyl group to the tyrosine residue whenever you have runs of C followed by G. And this is a very important phenomenon because it's known, for example, that if you have methylation in the promoter sequences of gene, then the gene expression will be switched off. If you look in the literature, most of the cancer epigenomics work to date have really mainly focused on tumor suppressor gene hypermethylation. But for our work, we're interested in this other phenomenon called global hypomethylation in cancer. So in other words, it has been reported that the cancer genome is more hypomethylated overall compared with a normal genome. And the reason why we're interested in that is because the hypomethylation is global, it's very pervasive. And we reckon that for our cancer test to work, the more pervasive a change is, the cheaper it would become to detect because you can sequence fewer molecules to get the same message. So first, we spent 2 years to actually develop a technology for whole genome bisulfite sequencing, which we can work in the plasma. And so basically what bisulfite sequencing does is that you treat DNA with sodium bisulfides. If the DNA is methylated, then basically the sequence will remain unchanged. If it's unmethylated, then the C will be changed into uracil, which when you do a DNA sequencing will look as a T. So first, we apply this whole genome bisulfite sequencing to the plasma of a group of 32 healthy control subjects. So we analyze our data in 1 megabase windows. And then we calculate the methylation density in each of those windows. So for example, if the methylation density is 80%, that means 80% of those Cs in the CpGs are methylated. So you can see from here that if you scan across the different windows, across the different chromosomes, basically it fluctuates a lot. And this is important because this means that there's a lot of interregional variations. So prior to this work, most of the people who look at hypomethylation in cancer have typically used quantitative PCR. So you can imagine if you just have one readout using quantitative PCR, you cannot capture that interregional fluctuation. But on the other hand, if you look across the 32 individuals, then actually the variation in the same windows is surprisingly low. In that, 95% of windows, the coefficient of variation is less than or equal to 1.8%. And this is important because for this test to work well, we need to have a very solid baseline, and we have that indeed. So next question is look at what happened in the cancer. So this is a circles plot in which I've plotted 2 circles in which the more outside a signal is, the more hypermethylated it is. The more inward something is, the more hypomethylated it is. So in the plot, basically here, the black one shows the buffy coats, the blood cells from this liver cancer patient. So you can see that by and large, the blood cells are pretty hypermethylated. So the methylation density is around 72%. But on the other hand, the purple one is the liver cancer tissue. So you can see the liver cancer is pretty hypomethylated throughout. So this confirms this global hypomethylation. But interesting, when you look at the plasma, you can see the plasma of this cancer patient is also very hypomethylated. And this is another patient showing exactly the same change. And in the early part of my talk, I show you that by sequencing a plasma, you can analyze the copy number aberrations. But here, in this part, I'm looking at bisulfite sequencing. So we're also interested whether bisulfite sequencing can also yield the copy number data. And so we'll take a look. Now, so in the innermost circles here is a tumor DNA using conventional non-bit sulfide sequencing. So you can see the copy number aberrations. In the middle one is the plasma using conventional non-bit sulfide sequencing. So once again, you can replicate the change that I see in a tumor. But outermost ring is the most important. This is a bit sulfide sequencing plasma DNA data. And then by the use of our algorithm, we can once again mine out the copy number aberration. And this is a second case showing that this approach actually works. So in other words, in one step, in one sequencing, I can mine both the methylation plus the copy number data. So I call this technology Magna. And then by use of this Magna approach, you can actually monitor the cancer patient. Now, for example, in one case, before surgery, in the methylation part, you can see there's a lot of hypomethylation in plasma. And then after surgery, most of those have normalizes. And indeed, actually, this patient remained well 20 months after surgery. And the copy number side of this Magna, you can see this is the copy number before operation. And after surgery, most of it has gone. And this is the second patient, where you can see that before treatment, there's a lot of hypomethylation in plasma. And interesting, after surgery, there's actually still a lot of residual hypomethylation. And indeed, this patient has a very stormy course in that he's found to have a liver cancer deposit in the non-resected liver, and later on developed multiple lung metastases and then passed away 8 months after surgery. And the copy number side is actually show exactly the same thing. This is a copy number before surgery, and afterwards you can still see those residual changes. So by having this Magna, you basically can combine methylation plus copy number at no additional cost. And you can combine them in different ways depending on how you wish to use the data. Now, for example, if you want a test which is very sensitive, then you wish to combine them by using an OR algorithm. So in other words, if either hypomethylation or copy number aberrations are detected, then you call that a positive result. But on the other hand, you might wish to have a test which is very specific, like if you're screening the healthy general population, in which case you can use an N algorithm, when only hypomethylation plus copy number aberration are seen before you call it a result as positive. So we've now actually sequenced 26 cases of liver cancer, or except one case is Barcelona stage A, so indicating that you can use it for resectable diseases. And the size of the tumor is from 16 centimeter all the way down to 1 centimeter, which is very small. And then most of the cases caused by hepatitis B virus, although there's one case caused by hepatitis C and a number which are non-viral etiology. So if you do the OR algorithm, then you have a sensitivity detection of 92% and a specificity of 88%. On the other hand, if you have an N algorithm, then you have a sensitivity of 69% and a specificity of 94%. And this is just a scatter plot showing the healthy subject, the liver cancer patients, and hepatitis B carriers. And we also show that there's a correlation between the hypomethylation we see in plasma and the fraction concentration measured by copy number aberration. And this plot is quite interesting because it actually shows that the hypomethylation is more sensitive than the copy number aberrations, because the detection limit using this depth of sequencing here, which is 100 million reads, is about 7.5% tumor DNA concentration in plasma. But on the other hand, the hypomethylation can go all the way down to 4%. And this is another correlation plot, which is a hypomethylation level in plasma correlate with the tumor size. And we've also further analyzed the data by using receiver operating characteristic curve analysis, or ROC curve. And this is the hypomethylation part. So just to remind you, a perfect test would be something which is just on the top left-hand corner with an area under the curve of 1. And a useless test will be one which is plotted over here. So basically, on this sort of analysis, you can see the hypomethylation is actually pretty impressive. The area under the curve is 0.93. And also because hypomethylation is so prevalent that I'm able to even sample just a tenth of the data, say 10 million reads, and actually the area under the curve is hardly changed at all. And this is important because the 10 million reads is the number of reads that we're now doing for those pregnant women going for prenatal testing. And we know that is commercially viable. So that we are talking about a test which is about 8,000 Hong Kong dollars. But on the other hand, if you look at copy number aberrations, if you look at 100 million reads, then the area under curve is 0.9, which is very similar to a hypomethylation. But on the other hand, if you only sample 10 million reads, then actually the area under curve has dropped significantly to 0.76. So indicating a copy number aberration is very sensitive to how deeply you sequence. And the reason for that is because the copy number aberration are focal. If you don't cover enough of those regions, then you just do not have the power. So this is interesting, means that if you combine these two together, copy number aberration and hypomethylation, the hypomethylation is probably a cheaper preliminary screen. So now those are data from liver cancer. So does this work for other cancer types? And so we'll look at a variety of other cancer types. For example, in breast cancer, this is a copy number aberrations, and this is a hypomethylation. And this is lung cancer, which shows the same thing. And this is smooth muscle sarcoma. And this is neuroendocrine tumor. And this is nasopharyngeal cancer. So basically, any cancer that we look at, we actually find that this technology can work. So those are the scatter plots in which is the controls. This is a non-metastatic cancer. So the red one is the liver cancer data. The blue one is the other cancer data. And this is the metastatic cases. So at a glance, you say, well, why is the metastatic cases, the signal seems to be lower? And the reason is because I'm plotting all the cancer together in this graph. But if you look at the data a bit more detailedly, then you can see that within each cancer type, actually metastatic cases generally have a higher level of hypomethylation in plasma. And this is a lung cancer, and this is a nasopharyngeal cancer. So it means there's a variation in the level of hypomethylation from cancer type to cancer type. But within each type of cancer, the more advanced stages still have a lower level. And this is the ROC curve combining all the data. So the blue one here is the hypomethylation, which once again shows that basically you can do 100 million reads or 10 million reads. It doesn't touch the effect for the methylation analysis. But for copy number analysis, you really need the reads to give you a good readout. And actually, this work was just published in PNAS in the early part of November. So in conclusion, hope I've outlined a technology which we call cancer genome scanning in plasma, which is a non-invasive approach for cancer detection, monitoring, and research. And I've shown you how one can do combined genomic and methylomic analysis, the so-called Magna. And this method is potentially universal and applicable to all cancer types. It's also a novel approach for studying tumoral heterogeneity. But clearly, at the moment, we have only sequenced 50 cases of cancer and 50 cases of control. So we need large-scale validation, which is ongoing at the moment. And currently, we can do this test for approximately $10,000 Hong Kong dollars. But I'll be very actively to see whether we can further reduce it. So finally, I'd like to thank individuals from my group for generating the data which I present to you, and many of them are in the audience today, and my various collaborators. So thank you very much for your time. Thank you.
Thank you, Professor Lo. So any questions from the audience?
Thank you for a wonderful talk. The results are amazing. So did I hear you right that right now you can do this roughly for $1,000 US, right?
Yes.
So this is for the tumor sequencing as well as the plasma DNA sequencing. Okay. So this just one. So about how many reads do you need for this to work?
Just for the plasma.
Just for the plasma,
yes.
Right. And
so the strategy which I'm thinking about is that because the methylation will work with 10 million reads, and whereas a CNA, you require more. So one very cheap way to combine is to do just methylation, so screen everyone with 10 million reads. If you give a positive signal, then we go deeper.
I see. So is there a difference in sensitivity according to the types of CNV? Amplification easier or deletion is easier? What do you think about it?
We have actually analyzed that, and as you would expect, it's related to the copy number, which is different. So if it's one copy gain and one copy loss, it's the same thing. But if it's two copy gains or above, then it's easier to detect. And by and large, you can actually calculate the degree of change. You can predict that relative to size. So for every twofold reduction in whatever you're detecting. You need 4 times as much read. Okay. Yes, there's a gentleman over there.
In almost all the data you've shown, you actually have the tumor sequences, right? So you have the plasma DNA sequences, but which are compared to the tumor sequences. So in absence of the tumor data, how easy is it for you to interpret the plasma data?
Well, I don't need the tumor data. I mean, the tumor data is only used as a gold standard to show that what I've shown in the plasma. I mean, I actually want to develop this as a screening test for cancer, so we don't have the tumor.
So in other words, if you detect hypomethylation in the plasma,
yes.
So which means that I have cancer of some type, but can I go and interpret those data as which type of cancer and the stage and so on?
We're working on that now. So basically what we've done is actually we look at the hypomethylation, and also we have some data in which we can even look at hypermethylation and the copy number aberrations, and put all those together for a hierarchical clustering. And that actually we can predict the type of cancer. So currently, for example, what we've done is that I've done the liver cancer versus all the other guys. So I can actually predict this is more likely this person has liver cancer rather than other type of cancer.
Thank you very much, Dennis, for the wonderful talk. Perhaps you can give the audience some idea what is the international competition and where are you positioned in this area?
Well, we're very aware of the international competition in this area. So one of our key competitor is Berk Vollstein and Victor Velasco from Johns Hopkins. So they have been looking at this area at the same time as we. Yeah, so they're one group. And then there's a Nissan Rosenfeld's group from Cambridge who also looking at this area. So, yeah, so we have to be very fast and, yeah, and make difficult decisions. So at the moment, I actually, because I think that in Hong Kong, one of the advantages is that we're very tight link with the hospital authority, and we have a very busy clinical schedule. So we can actually have a lot of samples coming in. So the next step for us is to actually use that for liver cancer screening. So we have this surveillance program, which Natalie has mentioned, and so we have already collected 1,000 cases of hepatitis B carriers who are screened regularly. And so I want to show that by use of this technology, I can predict which one is the one which will actually have hepatitis B early. Yeah, so that's our next step.
So, of course, you're aware that this has a very big commercial potential, and but this field is also, very competitive. So, do you have a roadmap on how to commercialize this, or what kind of help is needed from the Hong Kong government?
Over the last few months, I've been contacting Janet Wong, who's the Commissioner for Innovation Technology, and she has really been very kind to give us some advice. So we're actually going to spin out a company to try to commercialize this. And this is interesting because from the AOE work from before, I know a little bit about commercialization, but before that, I wasn't as aggressive. We just used a license out approach. But of course from that, it works, but I realized that most of the benefit actually go to the US instead of in Hong Kong. So for this time, we're going to actually have a local Hong Kong company. I mean, of course, that's risky, but I think it's a risk that I'm willing to take. Yeah.
I have a question here. So I can understand if you do the DNA test in the patient, in the cancer patient, then you'll find all these changes. But then if you look at the other way, if you find changes in the DNA, does it necessarily mean this person has cancer?
Yeah, so that's why when I did this work, I'm very interested in the specificity. The specificity has to be very high. That, for example, like when I use the N algorithm, I said that you can detect about 69% of the cancer, but at a specificity over, say, 94%. And you would say, well, 69 seems low, but on the other hand, you've got to bear in mind the current, those 69% of those people will prod along until many of them present with metastatic cancer. So to me, we're actually saving a proportion of those people. So that's why the specificity is important, because I don't want to have to do a lot of follow-up work. So over the last few months, we've been in discussion with many clinicians in our hospital about what should that follow-up test be. So currently, our conclusion is to actually focus it on liver cancer and to do liver MRI as our follow-up test. And of course, if you think it aloud later on, if I want to do a pan-cancer screening, then you might have to think about whole body MRI. But whole body MRI at the moment is still quite an early stage. And you might catch all sorts of other funny things. You might say, this guy has stroke or some other things. So we're still trying to think about the ethics. So currently, just work on liver cancer.
To detecting a tumor, do you think your method is better than tumor marker, such as a liver tumor marker AFP?
Well, I think AFP is not a very good tumor marker. I mean, this is quite poor, really, compared with this. And similar, PSA is also a terrible tumor marker. And CA, CA19. I think all of the tumor markers that are generally in sort of clinical use now are not comparable to this.
So if there's no more question from the audience, thank you, Professor.
Thank you.